Beleza, estamos transmitindo já.
Okay. Okay, thank you very much today for your visit. We appreciate very much. I myself had some work in Japan, Tsukuba, since 20 years ago. But nowadays I'm here in Paraná. I'm trying to have a team in genomics, plant genomics, which is very important for the future of Paraná because it's agriculture state, and we are looking for some new cultures of new genomes for chalk soybean, common beans, and soil metagenomics of soil. And you have this tool here. They are from the metagenomics microorganisms from soil. So these three projects together is what we call an agrigenomics project. And we're going to start next month the exhibition on. Have been working with the people together. My job is to put all these people together, find the people in genomics and plant breeding in soil and beans and soil and other cultures, crops as well. And we spend about 5 months putting people together and the arrangements. And now have the name of this new arrangement, new research and innovation and arrangement in agrigenomics, which is called NAPI. And it's a kind of way the foundation here can finance projects in here. So get people together. Good big projects with some expectation of results for the market, for the producers. It's we are linked into a problem, a big problem of the state, of the future of the state. And genomics and plant breeding in Brazil as a whole. We have very few people because of the transgenics plants. We lost many scientists in the United States, and so this project also has an aim of training people, young talents, and I think we'll be the first group in Brazil looking at genomes of plants. Name of plant breeding. And we have, as I'm going to show, many institutions of Paraná and some from Brazil as a whole, and looking for partnership outside Brazil. So we're glad you are here, we can talk, and maybe we can share some research together, aiming new plants. Okay. So the coordinators is more than people than that. It's just we have glaciers here in soil, and also Eduardo is here with coordinator in soil. Myself, I'm kind of a manager, project manager. Okay. It's a project, it's 5 years project, but we as a whole will be 16 years. It's a long-term project because it's genomics, it's genetics. You know? So it's involved 63 researchers and 9 institutions all over Paraná. See, I found very young people with good background, but no money for research. Because Paraná, before, in the years before, had no has not financed projects in research and in science, technology, and innovations. Okay? Nowadays, since the last year, they start with supporting these projects in science, technology, and innovation, focus on problems of the state, in health, in agriculture, and in areas to say. So there's many universities and institutions all over the Paraná that have been working alone. See? Now we are putting all these people together. And the idea is that the governor say that we have to produce many products because Paraná wants to be the supermarket of the world in terms of needs for food. And so we are looking for soybean, common beans, corn, not now, in the future, say, where it's much difficult at the moment. And trigo and cevada is for the future. But you see, we are very much dependent on soybean for animals and for cattle as well. So soybean is we are the second processing. That's the idea. The increase in agricultural productivity is out deforestation, but we have been doing very well in terms of productivity. See. Nowadays, so we want to go further. We want to do something else than just productivity. And we have to look at the soil because Paraná is a new state, a new agricultural area, and the soil here, the forest was to some decades ago, so it must be different from other places. And to have good plants, good crops, you have genomics and also the genomics of the microorganisms because of the health of the soil. And also, we have to ensure the benefit for agriculture and for other growers. Well, that's the main idea of the project. See. So focus on maximizing soybean and bean production through the optimization of wheat and the productive potential of the soil based on genomics. That's our goal. So we are going main products, a digital platform of breeding management, integrated with all omics technologies, and with focus on productivity and stress responses. As part of climate changes, we are going to need more resistant plants to climate. And also pest control in Brazil is a really big problem because of climate. And that's for the use of this research and also for companies will be 4 years, 3 to 5 years on basic science in genomics. And then afterwards, we're going to have more applied science. So Paraná is the second largest soybean producer in Brazil, so it's really matters for us. The families who have great involvement of families in producing soybean in Brazil, in Paraná, soybean, beans, and other cultures, all other crops. And also the main environmental factors, drought and high temperatures. So we are going to combine classic genetic breeding with genomic tools aiming to develop more productive cultures with greater speed and precision, tolerant to drought and high temperatures. And that's a predictive platforms for us. So, disease resistance and tolerance of pre-harvest sprout, and that's for, as I'm working with soybean, it's easy to work out so with common beans. It's made all sense to have more benefit for the producers. And Paraná is the largest producer of beans, common beans in Brazil. And we have institutions that is already working on plant breeding, but traditional breeding. Now we're introducing new technologies of genomics. So it's there's a delayed vacuum. It's a problem we're going to face. And disease resistance is a problem because the crop is so short.
Excuse me. The delayed seed coat darkening, it is when it's not black bean, it means that?
No, it's a key, sorry. It's some of our cultures, if you store them, they lost their original color. So lost value for the market. So I think we can help with some kind of technology and genomics to solve this coating delay. So that's it. Você quer falar pouquinho sobre isso aí?
Okay.
Soil, microbiome.
Yes, this project of soil microbiome is I think is the first of the world in this dimension, because we will map the whole state. It's about 8,000 samples, the whole state. And we have a lot of problems because we don't know what kind of microorganisms we have in the soil. And we are not only looking for microorganisms, but we are looking for antimicrobial resistance. We are looking for microplastics as well. And what we are trying to do is to combine the information that we have in the soils, with the metagenomic data, with the other data that are available as physical analysis, chemical analysis. We will map with drones as well, and we'll combine all these and try to find patterns with artificial intelligence, because our goal is in the future to create a platform where our farmers can put their information and have prediction of what will happen what will happen in the soil based on the culture that they are putting there, and based on the climate conditions, based on the location. So it's what we were trying to do in this project.
So that is very challenging because in the soil there are so many kind of the bacteria. So some bacteria is so good for example symbiosis or something like this, or something the pathogen, or something the kind of the good to what we say, good to condition the soil. So that is best challenging. But if we can find some genetic can find what kind of the bacteria exists, so we can to get more good data for the productivity and sometimes.
We are not focused only in the kind of bacteria, but all the genes that are present there, because we probably have a lot of genes that we don't know, and have a lot of bacteria that we don't know as well, fungi as well.
Yes.
So what is the pattern that we have in our soils?
So sometimes bacteria is easy to cultivate, but some other is not difficult, so that is okay.
I think 90% is not, we cannot cultivate it.
Okay. So the project have soil, as I'm seeing now, and genetic breeding, and genomic approaches, and in 3-4 years, we want to have this database and applications of genetic analysis and manipulation tools. There's CRISPR, all these new tools, and progressing from zero to some advanced.
So the CRISPR-Cas9, so Tsukuba University is a leading university, so it's not. So probably she knows.
Acho que acabou. So application of the products you have at the first beginning. So it's what I'm talking here. It's just the beginning of the story. You see. The basic research.
So when we got the project to start?
Next month.
Next month. So
we took about 4-5 months to get people together. More than that.
More
6 months.
No, I think it was a year.
A year.
Yeah.
Because of Paulo.
Yes, exactly.
A year, getting people together and get to know each other and submit a project to foundation. And they took some time and some questions, but now it's approved. We have a good budget, and we have what is in favor of us is this human genomics is file I had to say, and I have the equipments, the big equipment for genomics. So we're going to have the advantage of them. Okay.
Excuse me. There is 2 groups that work with microbiology, soil microbiology in Ponta Grossa State University. And I don't know if they are invited. Professor Gessele Batista works with inoculants, and Professor Carolina, and Galvão, and Rafael Eto. When you arrive in Ponta Grossa, there is one representative of this group that will take part of this meeting.
Okay. Well, what happened is, the situation we got here is many people working in parts of the store, you see, and we got together some of them, not the whole people, and submit the project. Now it's approved, but we know that we have gaps, see, to be filled up. But at the beginning, we thought that if you try to find every person state, it would be a bit difficult. So we have a good project now, it makes sense, but there's gaps, see? And we know that there's more people in the state able to collaborate. So after the approval, which is just came, we are going to start. So now a new stage of the idea, and to talk to each other, see the gaps you have, and find in state, in Brazil, and outside Brazil as well. Because it's not easy. It's not an easy project. It's so many specialities, many people involved, and to get a good result, it's not easy. But we're going to see other people. We have mapping some of their people that's working. And sometimes many of these people are working in productivity, you see, which is not, it's not our main aim and goal.
This is general.
At genomics, we can at the end, at the very end, we have to provide the cooperatives, the multinationals, with some clues what the genes are able to produce to help in new plants. The idea is a new soybean, new chromosome beans. That's the
So the target is soybean and common bean.
Why?
No. Just want to know in this project you are targeting on that's two kind of the crops.
Okay.
That's all.
Soybean and common bean.
Feijão, yeah. Common bean.
Feijão, because it's too close.
Yeah, it's close.
So will be instead of corn. Corn, maize will be a big challenge.
Yeah, different, yeah.
We are going to get there, but we have to train people first. And soil, which is nowadays soil is tremendously important.
So that project is Paraná state funded project.
Yes.
So that's why that is all the
Yes.
researchers in the Paraná.
There's no federal money here yet.
Yes.
And there's no similar project in other states in Brazil. Even in Brava. Brava is coming inside here. Brava here. Because what happens with research in Brazil sometimes, it depends on the area, but in agriculture, you have many researchers doing small things in different places for publication.
Exactly, yes.
Not for results to the society. That's different. Now it's different. We're doing different things. Publication, like you do in Japan.
Yeah.
Publication will come after, not at the beginning. It's a stage of the evolution of the way you produce science.
Yeah.
Because Brazil is quite a new country in terms of science and technology. So. Okay, want to say something about your,
I have one question. So you focus on the microbes. Yes. So that you also focus on the regular lines of your interaction.
Symbiosis.
In this moment, no, because we cannot, we thought about the interactions, but it's a lot of things. So in this first moment, we will map the soil, but we will map what crop is in the soil in that moment, but the interaction this moment, no. Probably we'll have small projects coming from this. But
yeah, that's the idea.
yes. And probably in the future, we will take these microorganisms and isolate the microorganisms, but the first moment, we'll just map.
Okay, the first stage just survey.
Yes,
From using with the genetics.
yes.
Okay, I see. You're quite interesting project.
Yes, and I'll just put my email here and I'll tell you. It's a really interesting project. We are really excited and we are really open to collaborations. It's a huge project. We have a lot, as Professor Evaldon told us, we have a lot of gaps and we are trying to do our best. Eduardo is responsible for all the IT things, the EI things, so we can talk a little about this.
The idea is to merge all this data and extract profile
But some,
of the soil characteristics from the different places. And we think that this profile will show us what we need to do in other projects to improve productivity, for example. This is one of the goals, just one, but the idea is to use this data to extract other information. For example, what the microbial resistance genes and other kind of stuff.
do you want to show us what you are doing in Japan, your research? Is it possible that we can know what you are doing?
You do the presentation.
You gotta connect the internet.
You speak some Portuguese?
Bobo.
Ah,
So, can I connect in the internet? But can I do it?
you can connect the internet.
Can I do it? Can I?
So that in future, the next stage, so you will focus on the nutrition of the soils, not only genetics.
In this moment,
In moment,
Already
because the idea is to merge all the data, all the metagenomics data with chemical and physical analysis, the soil, with the merge with the kind of crop that is growing there, and for example, what the producer did with the soil in the last
season.
season. The idea is to merge all the data that we can think related to that sample point, and then we can, the idea is to extract from this amount of data information that is relevant for productivity, for example. The basic idea is to obtain a profile of the best soil and property that we have here.
And the best and the worst.
The worst, yeah, the idea is,
That one is important.
the idea is
Yes.
in each property, we will collect 2 sample points in each property. And one sample point is from good soil, and the other sample point is from bad soil, because the farmer usually knows in the property where is the most productive
area and where is the less productive area. And then is to check what happens in one area in contrast with the other area.
As well in the whole,
Yeah, and then we expand to a region and then we expand to the state, because we think that some patterns will appear here and there, and then we can assume or conclude that this is really relevant or useful, or we should do something.
yes, and
But it's just the big picture, right? It's just the main idea.
So I just little bit introduce our institute, JUCAS. So our JUCAS, the Japan International Research Center for Agricultural Sciences. So we are not only focused on the agriculture, but also the forestry and fisheries. And
Where you are there in Tsukuba?
yeah, that is Cuba. Yes, and that is our main purpose is collaborate with the developing countries to undertake the comprehensive experimental research and technology advanced agriculture and tropical and subtropical zone and developing countries. And to collect and analyze and research also and publish information on domestic and international research. And to invest and provide manpower and technical support. So for example, I went to the Embrapa Soja for collaborating like this. So the support the business and research to utilize the result of experimental research. So that is our main objective. So and because I just would like to introduce the new project that is the Green Asia project, but that is Asia project, but not, I'm sorry, that is not South America, but that is a good, for example, because
Monsoon, where is the?
monsoon, that is a monsoon region. Monsoon is that is the name of the wind. So the typical wind in Southeast Asia and Japan also. So we are collecting our research results to expand over this Southeast Asia region or Asian monsoon region. So we can put on our own research technologies to like this, show like this, and if somebody interested on this technique or this technology, so you can see, for example, the prolonged mid-season drainage paddy fields. So that is the kind of I think south part of the Brazil, for example, Porto Alegre have some rice cultivation, paddy rice fields. So for example, we can click here, so we can see the background and the results. So if they want to use this technology, you can so just click here and you can contact us. So that is kind of the first step of the collaboration. So that is we can put so also we can find the catalog like this. That's it. So now we collect not only our institute, but also another institute we call NARO, that is a national institute, but that is focused on the Japan region. Also, that technology catalog includes some another institute, also the universities, and we can make catalog like this. So you can download this in our web page. So that is the Asian region, and also we make this technology showcase for sub-Saharan Africa. So now we already completed the Asia and Africa. So next is South America. Yes. So now our GIRCAS is thinking about the next project with the South America. So if we can find the good technology to apply the South Africa, we will make this catalog. So somebody want to use this catalog, this technology, so we can use. So that is our new project to not only the research, but also that's the disseminate our technology and collaborate, new collaboration like this.
For the benefit of producers.
Yes. Not only producer, but researchers also and the policy maker also.
Okay. Of course.
So that is the introduction of our not research, but research results or something like this.
Good. Congratulations, good work.
So in this, we have the 5 years project, and this year we finished on this. Not Africa. Africa is previous, the first, and the second is now we are focused on the Southeast Asia region.
And
So,
do you have an idea or plans for South America?
yeah. We need to make a new project because we need the first step. We need to make some proposal and apply to the institutes under the Ministry of Agriculture, Forestry and Fisheries. So that's it. If the ministry approved our research, so we can start. So and now we are making the new project.
Okay.
Yeah, and we also thinking about the research with the South America, especially Brazil. So but until now I couldn't say we can start. I'm not sure, but we have tried to apply.
Okay. And which institution in Brazil? In Embrapa?
No, we didn't decide yet, but that is not decide envelope soldier, but that is, I can say that is the whole catalog. So I mean that if we can focus on the Brazil, so not only Paraná but also the another. So because the tropical region is not here, but
Okay.
Yeah. Okay.
Tropical.
Yeah. So
I need to try something.
Ele não tá usando isso. Não.
Não? Qual que é?
Deixa aí.
É que eu tenho que passar no.
É porque tá tocando entre as abas. Não vai adiantar você projetar isso.
É que eu tinha que colocar na live.
Mas-
So I would like to know if we make this catalog in Brazil, so that is everyone want to use one.
Okay.
You want to use one?
Oh, yes,
Yeah.
I think so, yeah, we're very happy.
Yeah, so that is not only the breeding, but also
Okay,
the,
okay, indeed. Information,
yeah.
data.
Yes.
Escolhe janela. Ou tá cheio.
Aí agora consegue
Uhum.
passar aqui, vai estar.
Isso.
Beleza.
Yes, that's all.
Okay, thank you. And you was the same institution?
No,
No,
No.
your institution in Japan, which is, you work with.
I work at the University of Tsukuba. And
you can explain a little about your job there.
My job here, okay. So that I previously introduced you that presentation, you remember?
Yes, remember,
But
the
that-
online, yes.
Online, so
Oh,
the,
yes.
yeah,
new tomato line with high nutrition of GABA. You know the GABA? It's an amino acid. One of the GABA is good for the relaxation and low blood.
Stress that.
Yeah, yes,
Yeah.
that's right. So the Japanese people like the GABA. That's why I try to.
Not just-
Yeah, another one is melon.
Hello.
Melon, that he's a professional. So melon, the Japanese melon is quite expensive. You know how much is it?
No.
About the luxury one is around $300, something for one.
300?
Maybe.
300 what?
What?
Dollars.
Dollar.
For one fruit. Yeah, it's a special present.
Maybe, okay. We need to
Go
wait
to
to
the-
go to the supermarket.
Let's go to the supermarket.
Sometimes Japanese people will be too crazy.
São loucos.
Yeah, very,
If
very
you want to go to the supermarket later,
No, we can buy that luxury one. We can buy the special store.
okay,
Mhm.
okay.
So the
But they cultivate
oh
an apple in Japan,
oh
in stufas, in greenhouses.
so the
They lack apples.
We too.
But
And it's Brazilian.
but some Brazilians don't like it.
The
They
acid
100%
pineapple.
love it.
And this, and
Brazilian people don't like acidic pineapple.
Yeah.
yes, yes.
Why? Good.
Capixaba, and
and
abacaxi. Vale só abacaxi.
Pink now.
so then
Depois eu vou contar para vocês, eu conheci o cara da Mossoró que exporta a melancia da Japão. Tem um caso muito interessante, vou contar para vocês depois. Mas engraçado, ela estava lembrando de mim, que eu estava olhando a foto que ela apareceu online. Apareceu só muito mais velho. Online. Eu não estava reconhecendo que era ela. Ela não apareceu.
Sim.
Xenofóbico.
Pode ser. Pode ser bem velhinha.
so the my
Uma moça.
yeah my
Ela trabalha com genética de planta e de peixe.
De peixe. Ela trabalha com CRISPR, que nem a Tassi vai trabalhar.
so my oh oh what oh how should I say what are you doing no sorry
Vou na câmera pra
this one now is okay
Isso.
yeah it's okay
Yeah.
oh so I interested in about the plant transformation because the transformation is a very important technique for analysis of gene function
Ok,
and the plant for molecular breeding.
ok.
That's why I'm very, very interested in this technique. So the plant molecular breeding includes the GM crops and the genome editing crops. Yeah, the transformation includes a direct method and agrobacter-mediated transformation. I focus on the agrobacter-mediated transformation. My career is study about this. Skip. So because of this process, but the problem is that not all crops are acceptable for transformation. So moreover, even the same crop, some cultivar are acceptable for transformation, but others are not. Yeah, especially melon.
Melon are not.
Yeah. But he's a specialist. Yeah,
I try.
I study his paper.
Oh,
Yeah, I'm very happy because his paper I read many times. This today I know he is a writer. I'm very happy.
You
Yeah.
like that?
When I student, I read many times his paper.
Really nice.
Yeah.
Good, the man was up all around.
You made good things.
Yeah. In some crops, in the difficulties of cultivar makes the transformation difficult. For example, rice, wheat, maize, soybean, melon, etc., etc., etc. So what of genetic factor does make the transformation difficult? This is nowadays my question, interesting question. So my final goal is make all crop acceptable for transformation. It's my dream. And so for this goal, to go to this goal, I have a trial to modify the agrobacterium. So the agrobacterium infected to plant cell, the plant cell make the ethylene and GABA. These two things is avoid to introduce the agrobacterium because these two things are defense response from the plant. That's why I introduced these two. I introduced to agrobacterium the ability to avoid the ethylene evolution and GABA degradation. Yeah, this is my first trial. So I introduced two enzymatic gene. One is the ACC deaminase, and other is a GABA-T, GABA transaminase. The ACC deaminase cleavage of the ACC, the precursor of ethylene, you know, and the GABA-T is a degraded GABA. These two are the ACC deaminase from Pseudomonas and the GABA-T from E. coli. Cotyledon. Then 3 days after co-cultivation, the control is like this. Only ACD, agrobacterium is only ACDS, only GABA-T, and both. But these are increased compared to the control, like this. This is the same. We test this agrobacterium for the melon, white watermelon, eggs melon, tomato, canola, that increase the transformation efficiency. My supervisor commercialize this tomato. I'm happy to hear that. And next is a long shelf life melon with Prismatas 9.
You use the promoter, inducive promoter or a promoter constitutive?
Constitutive promoter. Yeah, I always use a constitutive promoter.
Okay.
It's I forgot the question.
Excuse me.
Yeah.
When you use CRISPR-Cas9, you can extend shelf life.
Yeah.
But you need to put in the agrobacterium to introduce into the plant and then rescue the transgenic plants.
Yes. Yes, and in tomato, The first T cell generation includes the CRISPR-Cas9 system in the genome, but the next generation segregated out depends on the Mendelian law. We select the line, no CRISPR-Cas9, only mutation. New line.
And it takes how much time to do that until arrive to the new plant?
1.5 years, 1 year plus 6 months in tomato case.
In melon case.
Melon, because the tomato is become 2 generations per year, but melon is tsukuba is too cold.
It's on field. It's in the field or greenhouse.
Greenhouse. Even I use the greenhouse. Tsukuba's winter is very, very cold. We cannot do 2 generations for melon. Tomato is okay 2 generations, but melon is 2 or 2 and 6 months in Japan. Spain and some area is shorter than. I think Brazil is shorter, yes, for the generation.
Yeah, but in Ponta Grossa, I do only 1 generation
Oh, really?
because the temperature is too much.
But in the northeast,
Exactly, yes.
3 years for 1 generation.
In the northeast, in
The northwest.
Ceará, Salvador,
Rio Grande do Norte, Mossoró,
far from here.
Nordeste is northeast.
It's a warmer place.
Well,
Good.
Plenty of light and warm and water.
Okay.
irrigation.
Yes.
But tsukuba is not good for tomato. Tomato is okay, but melon is not good for. But I like melon, so I have an allergy for melon because I do the pollination.
Yeah.
I can't eat melon, unfortunately. So okay.
But you make money.
Yeah, so but this melon is a Japanese luxury melon.
My God.
Yeah. Yes. Yeah. That's all my presentation.
Because you work with fish as well. Fish you work with.
Sorry, this
Not
is not
anymore.
no, because I'm plant scientist. Yes, so sorry, I'm not animal scientist.
Okay.
I'm not fish scientist. yeah, Professor Watanabe ordered me
Okay.
to present about the animal situation.
Okay.
That's why, that's why I present you at the moment. Very sorry. So
President will speak on
yeah, you are very interested in the fish.
No, because in here, Toledo, for instance, there's tilapia production. Tilapia.
Tilapia.
Tilapia. But it's, I think it's the main center of production of tilapia in Brazil. Toledo.
Toledo is one of the hugest, I'm not sure if it's the biggest, but it's one of the biggest producers of some feeders.
Aquaculture.
Aquaculture. But tilapia.
Yes.
Not paku.
No.
No.
Brazil,
Dourado.
we lack river.
Yeah, river here.
River.
We're very poor.
Yes.
Tilapia is nowadays the
Main
fish for river fish
river.
for the whole Brazil. Amazon is different.
Yeah.
High water.
Yeah, another.
But in here, The only fish you have is tilapia.
Yeah.
Which is very good.
I also like tilapia. In London, sometimes we can eat the sashimi tilapia.
Yes, sashimi is tilapia. Here, everything with tilapia. It's cheaper than salmon.
Yeah.
Professor, unfortunately, we need to walk.
Okay, but it was very good to meet you.
Yeah.
We have your interests, you have mine, our interests. As I said, we're just starting.
Yes.
Having the money for the people is now the researchers are now knowing each other, you see.
Yes.
So I think in some time in the future, but still this year, I think we're going to look for collaboration and very happy to contact you. If you have interest, we can work in some projects.
Yes.
We have in agriculture, we have been partners of Japan and Brazil in agriculture research, and it's very successful. So we appreciate if we can work in some areas. Okay?
Okay.
Thank you.
Okay, thank you.
Have a good time in Brazil.
Yeah, thank you very much.
Thank you so much.
When you are going back to Japan?
The next Saturday.
Okay.
Certainly.
This week.
22 this week. 22, we take a flight.
Because if you could stay more, one week more, we're going to have a meeting.
Yeah, I hope so, but Professor Watanabe say, we should go back. I hope so, but.
We have a meeting on 27 and 28 of plans here in Toledo.
Really?
Yes.
Yeah.
Yeah, it's a pity.
But they can have now the invitation to come back to see us.
Yeah. Well, I think we keep in touch.
Yes.
Thank you for being here.
Thank you very much.
Thank you so much.
Obrigado,
Obrigado. Obrigado.
Tem acabou aqui a reunião, né? Deixa eu voltar para esse
Ricardo, foi um prazer, eu tenho o seu contato aqui. Tá bom. Tá bom. Eu tenho